So continuing with table 6.2. All right, so a phototroph. Can you see that at all? There we go. There's a laser pointer in the middle. All right, a photoautotroph. Photo light auto self. Okay, photoautotroph uses its energy source of sunlight, and it uses carbon dioxide as its carbon source, so from the air. These are going to be photosynthetic organisms. Things like, well, probably the one that you would be most familiar with would be plants. Plants use the sun and carbon dioxide out of the air, and they make their own energy from that. Okay. Um, Algae are another real good example of this. Algae are green because they are photosynthetic. Cyanobacteria. Cyanobacteria also have some type of chloroplast variant that allows them to use sunlight. Now, a chemoautotroph. Remember, chemo is chemical. So, as far as an energy source, it would be simple inorganic compounds. Okay, so it would be literally a chemical chemo that they're getting their energy source from, but they are still getting that CO2 or carbon source from CO2 from the air. This is going to be certain bacteria, such as methanogens. And remember, I talked about the extremophiles in Archaea who live in the deep sea vents from basically volcanoes. Okay, they're going to be part of this group here. Okay, now those are the autotrophs. Let's talk about the heterotrophs. Okay, a photoheterotroph. Photo means light, right? So the energy source is going to be sunlight, but the carbon source is going to be organic. They're going to have to consume a sugar, a lipid, a protein, a nucleic acid. They're going to have to get it from one of the organic molecules. So examples of a photoheterotroph are going to be purple and green photosynthetic bacteria. Literally, purple or green photosynthetic bacteria are going to use wavelengths of light to get their energy. Okay. Now, a chemoheterotroph. All right. Um, this is going to be um, consuming some type of nutrient and then doing chemical reactions, metabolic conversions, to get the energy out of it. We, as humans, consume fats, we consume sugars, we consume protein, and our body uses enzymes to break it down into a usable source of usually glucose. Glucose is kind of the most common that allows us to put that into our energy producing um, process that we can break down to get energy or make ATP from. Remember, ATP is our fuel. Okay. So ATP is going to be like our gasoline. Um, Gasoline is not fire, right? ATP is not energy. It's not the same thing, but we can burn through gasoline to get fire. We can break down ATP to get energy. Okay. So again, chemoheterotroph, we're talking about the energy source being we consume a nutrient and we break it down to make it into something useful. As far as the carbon source goes, again, it's going to be something that we have to consume. We're not going to get it out of the air. So chemoheterotrophs include the protozoans, the fungi. A lot of bacteria work this way, and animals. Some animals work this way as well. Well, actually, all animals work this way. I don't know of an animal that doesn't consume food. So there are two types of chemoheterotrophs, okay? Saprobes, right here, or a parasite, right here. So a saprobe, okay? These are metabolizing the organic matter of dead organisms. The carbon source is still organic. I'm still consuming something. I'm still eating something. The two primary saprobes are fungi and bacteria. Remember, we talked about fungi being decomposers. They're going to land on something that's dead, and whether it's leaf matter or an organism, and they're going to decompose it. They're going to break it down. Bacteria do the same thing. And the thing with bacteria is normally they also produce gas. So I know this is gross, but since I commute back and forth to Kingsville, if you've ever seen a dead animal on the side of the road and it looks like it's blown up like a balloon, one of the reasons that that has happened is because the bacteria that are inside of it have started to decompose the inside. And while doing it, they have started to produce gas. As that gas gets produced, if they're internal, if they're actually inside, there is no exit for that gas. So the animal kind of blows up like a helium balloon. Okay. But again, this is decomposition and it's the bacteria that are doing it. Now, parasites, they utilize the tissues, and fluids of a live host. And again, because we're consuming, our source is going to be organic. Okay. There are various parasites that work this way and pathogens. It can be bacteria, it can be fungi, hello ringworm, protozoans, or animals. Some animals are parasites. Okie doke. Okay. So, free livers versus parasites. Okay. So, the saprobes. These are free living organisms that feed primarily on organic detritus, which is basically dead things from dead organisms, the matter that's left from dead organisms. Um, they decompose plant litter. Ugh. That's really hard to say. I don't know why, but it is. Animal matter and dead microbes. So we talked about the fact that, fungus a lot of times lands on things, let's say on a forest floor, and it helps to break them down so that it doesn't stay there forever. Now, why is this important? This is why. They're very important for the recycling of organic nutrients. Okay? Remember that if things didn't decompose, all of those organic materials, all of the proteins and carbs and lipids and anything else that was in the materials, let's say in the dead leaves, would just stay there because it wouldn't get broken down. So having that happen is really important. Or in a forest, you would have, you know, 20 feet of dead leaves because nothing would ever break it down. Okay. Parasites, however, are going to derive nutrients from the cells or tissues of a living host. So here, it's already dead. We eat it, no big deal, it's already dead. Here, my host has to be alive. Okay. We've got pathogens. These are parasites that cause damage to tissues or tissue death. We've got inside, cellular parasites. These guys live within your cells and basically thrive inside your cell. And then we've got obligate parasites. Basically, they can't grow outside of a living host, they have to be there. Um, okay. Yeah. So again, saprobes, detritus, dead things, parasites have to have a living host, and there are different types of parasites. And the big thing with the saprobes is they're important recyclers of organic nutrients. All right. Environmental factors that influence our microbes. Okay. Multiple environmental factors affect how microbes live and how they grow. They include things like temperature, oxygen, pH, which is it acidic or is it basic or alkaline? Um, Osmotic pressure, how much water is there? Radiation. Radiation can even affect you and me, right? We don't go around going, you know what? I just want to get exposed to radiation. No, you don't. Relationships with other organisms will also influence how those microbes survive. These environmental factors can greatly affect the function of microbial enzymes, which can make or break the functions going on in the cell. Depending on how, let's say, temperature sensitive a microbe is, you may only find it in a very specific location because that is the only location that actually has that temperature available to them. Okay? Most organisms must live within a narrow range of environmental conditions in order for their cellular processes to operate uninterrupted. Um, let's see. All right, so I'm sure in some biology class that you've had, you've heard of homeostasis. Homeostasis probably was taught to you as a set point. The thing is, homeostasis isn't just a set point, it's a set point suitable to support life. When I talk about the window is a set point suitable to support life. There is a temperature window suitable to support life for bacteria. And for us, honestly, we do well at 98.6, but we also do well at 97.6. There's a window there. We can live with a temperature of 102. We're not going to like it. It isn't the best place for us to be, but we can live there. Same with 103, same with 104. Get to 105 and 106, and bad things start happening. And we're getting right at 105, we're right at the edge of that window. By 106, usually you're starting brain damage. So, again, that temperature window that we are safe in, for us anyway, as humans, is pretty wide. Bacteria, microbes usually don't have that big of a window, which is why our body likes fevers when you're sick. Because if you've got an infection, microbes don't like different temperatures. They like 98.6. So when you're not 98.6, they're not happy. Now, does that mean that they won't survive? Sometimes, not always. Sometimes it just means that you're slowing down their growth. So they're not able to thrive in your body. Okay? So let's talk about temperature. Cardinal temperatures are the range of temperatures a microbial species can live and grow under. You've got a minimum temperature, which is the lowest temperature that permits a microbe's continued growth and metabolism. Below that temperature, cellular activities are severely limited. You have a maximum temperature. It's the highest temperature at which growth and metabolism can occur before the proteins are denatured. See that? Denaturing a protein basically means that you unfold it, you break it down. So, the best example that I can think of, because people will say, well, what if it cools down? Will it go back to the original shape? Think about having a knit sweater. Okay. I knit and I crochet, just so that you know. Let's say there's a string and you start pulling the string and the sweater starts to unravel. And you start unraveling that sweater. Can you put it back together again? I knit and crochet, and I don't think I could. So when we talk about denaturing proteins, when we talk about unraveling or unfolding the proteins, that's like taking apart the knit sweater. If you do that, that protein is damaged and it's not going to work anymore. Now, do it for enough of the proteins inside of that cell, and yeah, the cell's not going to be able to do anything because all of the machinery inside the cell that allows it to function is now in pieces and it's fallen apart. Okay? So, again, maximum temperature is still okay, but you get past it and dangerous territory. In the middle of minimum and maximum is usually the optimum temperature. The optimum temperature is between the minimum and the maximum, and basically it is the absolute perfection of temperature that allows them to grow the fastest that they can. So, the best way that, well, this is how I learned it. If I were to have a graph, okay, with time on the x-axis and temperature on the y-axis, okay? There is a high and a low point. This would be the max temp. This would be the min temp, okay? And then dead center in the middle right here is going to be optimum, okay? So this would be homeostasis. All right? It would be absolute perfection. This is where everything is aligned so that I can make copies of myself as a bacteria as fast as I possibly can. And as long as I am in, okay, as long as I'm in this window, I'm going to survive. Like you with a temperature of 102, you don't like it very much, but you'll survive, right? So if I'm in the window, top or bottom, max or min, I'll survive. But that one line in the middle, the optimum, is when I'm my best bacteria. I'm living my best bacteria life. Okay? It gets dangerous though if I go above this or below this. That's when things start going really, really wrong. So for a human, that would be us getting to a temperature of 106. Because at 106, you start unraveling your own proteins and people go into seizures. People start, again, seeing brain damage. So that window between max and min is where, as a bacteria, I want to be. Okay. Pause for a sec. All right, so here are examples of temperature ranges for specific types of bacteria. So psychrophiles, the blue line here, okay? They like it between negative 20 and 15 degrees. They like it cold. Remember I talked about the bacteria you find in icebergs? They like it cold. Notice that when you get above that 15 degrees, there are no blue lines. That's because I'm outside of the homeostatic window. They must live in cold temperatures to survive. They're obligated to be at cold temperatures to live. Okay? Um, psychotrophs. Psychotrophs, the second bullet. Okay? This is the orange. All right? Psychotroph grows slowly in the cold, but their optimum temperature is usually between 15 and 30 degrees Celsius. Okay? So again, you can see where they're growing, but it's not that great. They're growing, but it's not that great. But their perfect temperature range is usually 15 to 30. That's where the top of that arc is. Okay? Mesophiles, this is the green. Mesophiles grow between 20 and 40 degrees Celsius. The majority of our medically significant microbes fall into the category of a mesophile. They basically are within our body temperature range, okay, where we do well, basically. Thermophiles, that's these guys right here. Thermophiles grow best at temperatures greater than 45 degrees Celsius, um, usually to about 80. And then the extreme thermophiles, we're talking about probably 67 degrees Celsius to about 130 or above. Okay. Again, not going to be the best growth, not going to be the best growth, but they can survive it. They're still within that window. So these are the guys that you find in the hot springs in Yellowstone or volcano vents. They're the ones that are, again, obligated to stay at those temperatures. They normally don't do well when you take them out of that window. Okay.